Right. Today we are going to have a look at um, the real um, physiological experiment. Well, it's not only physiology, it's the combination of other disciplines as well. But I think it's a good way to show you how it's done. You see, you have um, learned a few things about physiology. And some of them are very fundamental, some of them are a bit um, advanced. So these knowledge actually are required, especially when you want to design your experiment, you want to um, decide how you want to test your hypothesis, what kind of treatments you want to give to your plants, and then when there are responses, how do you want to quantify these responses? And also, how do you analyze the responses? Okay, and then after that, of course, there will come the discussion to make sense about everything. Okay, so for now, I've taken this one paper about the physiology in rice. Okay, it's quite a recent paper. It's published in mid of 2018. It's about rice, how rice are able to live aerobically and also anaerobically. What happens to the rice that will make it um, able to live in two different environments? So aerobic rice, it's rice that do not live in the regular flooding condition. So it's pretty much the land which is rather not to say dry, just optimum moisture. For example, like the upland rice. Okay, if you go to the Borneo, you can see people grow the what's the name of that rice? Barrio rice. Yeah, that kind of rice. Upland rice. It's under the category of aerobic rice. Anaerobic rice is the conventional regular kind of rice that you always see in the field. It's flooding condition. It's very muddy. You need to use a lot of water to do this kind of cultivation, and so on. So um, it's I will go to each of this section pretty slowly so that you will have time to appreciate the content of this. Okay. Okay. 1 second. Why this is not moving? I have to close this for a bit. All right. That's why. Okay. Why this is important? Um, you know, it doesn't matter so much um how much the physiology books you remember, or how good you are at doing your experiment. If you fail to report your finding and then making a good sense about it, and then you let people know about it, it's pretty much useless. Okay, so you need to come up with some good understanding, not only how to do your scientific experiment, but how to convey the finding that you have just made to the rest of the world, of course. And to do this, your discretion, um, I don't say that it has to be like super English kind of proficiency. Not really that. It's more into like how structured you can get when you want to present. Okay, so this is a regular paper. So I suggest you from now on, um, you should be, because some of you are doing final year project, some of you are master students, you need to be able to become familiarized with this kind of articles. Okay, this is journal article, research articles. Okay, so let's get started. So for scientific article, there will be a title. Okay, right? So there will be a title, and the title for this article: studies on root anatomy, morphology, physiology of rice grown under aerobic and anaerobic condition. So in titles, you are going to find things like variables, of course, variables. Okay, so there are three variables that should be present in the title. It should be the constant variable. You need to have the manipulated variable, and also your responding variable, the things that you measure. Okay, so the constant variable, what are the constant variables here? Do you know what do you think? Yeah, you're talking about, to some degree, you're talking a lot about rice. So this can be your constant variable. What do you manipulate in your study? It's the aerobic and anaerobic condition. Okay, so this will be the things that you manipulate, hence become your manipulated variable. Okay, and then what about the responding? Responding, the things that you measure. How, after you have treated your plants, what change? Okay, so things like root anatomy, morphology, and physiology. So these all now will become your responding variable. So I would say that this is a good title. Okay, just bear in mind as we go along down the line here. Uh, Scientific articles, even though it has to be superbly scientific, it is still written by human. So you might see from time to time there will be errors. Okay, so a good scientist, when they read a journal paper, not only that they understand the journal paper, they can make a good constructive criticism. Okay, for example, after they have read some portion of the journal, the article, they can make a suggestion. Okay, not a disruptive suggestion, a constructive one. This should be done this way because it is much better. Okay, so this paper is also included. You will see that there are things which can be done a bit better. All right. Remember, nobody is perfect, but we practice to make it more refined from time to time. Okay. Right. So we got our title. So that's good. Title. Oh, by the way, I forgot to tell you. To do all of this, we actually have this format. Have I shown you this? I have shown you this. Yeah. So this is the scientific format actually. How to do any articles. You should have these components. You should have the title. I'll just put it right next to here, okay? So that you can see. You should have your title, abstract, keywords, which we have here. Yeah, we got the title, we got the authors, and we need to follow by introduction, method, result, discussion, conclusion, and also references. So regardless of any scientific finding that you want to write, you should be following this IMRAD format. Okay, must include all these components. You're not going to be wrong about it. All right. So we put this aside first. I can give you this in a bit. Maybe have I given you this? Anyway, I'll just put in your chat. Yeah, I'll just put in your chat. Okay. All right. Okay. Then you will also have an abstract. Okay. So let's read. I told you, right? This is going to be a bit slow, but I want to get it right with you. So it says, with the changing climate and rainfall aberrations, there is a gradual shift in the system of rice cultivation from traditional transplanted anaerobic to aerobic system. So it just tells, it just tells you about um, the general information about a rice cultivation system. Okay, from anaerobic to aerobic system due to this rainfall pattern. Okay, and therefore you will see that this is written in present tense. Present tense, yes, you can use present tense in abstract, not past tense all the time, if you are talking about a general statement, something habitual. Okay, just like this statement here. And then what it says, studies on the root anatomical and morphophysiological traits provide insights about the adaptation under aerobic condition. Again, this is present tense. I'll just use this as a abbreviation. Okay, so present tense. Oh, sorry. I'll just put R. If it's past tense, I'll put, okay. So PRS is for the present. Past tense is for the past. What about the future, if any? Future tense. Just want to. So future tense, I will put PFD. Okay. So this, the second one, is still present tense. Present tense. Okay. The third sentence, we investigated the root anatomical and morphophysiological traits in anaerobic BPT 5204 and anaerobic CR done 202. So this is rice variety. Okay, you see, scientific article is quite advanced. So very often you need to have some kind of pre knowledge about various things, not just about science. Sometimes just about other things as well, because you can't see like a movie how the scientists did their work. So pretty much you need to imagine things as you go along. So please, this you need that skill, active reading. As you read, imagine things, make the connection, make your brain work a lot. Okay. So we know that BPT 520 rice variety, which is anaerobic. So we can write over here BPT 5204. This is anaerobic adapted rice, and we have CRDHAN 202, which is aerobic rice. How to spell it? AEROBIC, aerobic rice. Okay. One thing that you need, if you're not familiar with rice is able to both grow in anaerobic and aerobic condition. However, because it's a semi-aquatic plant, um, after some time, it can adapt to the situation or the condition, the growing condition. So that's why we have this BPT, we associated with anaerobic adapted, while CRDHAN 202, we call it to be adapted to aerobic condition. Okay, right. It was observed that the formation of fewer parenchyma, thickened root, and larger xylem area were critical anatomical trait associated with aerobic adaptation as compared to anaerobic condition. Okay, so parenchyma, please, this is a type of tissue. Okay, air parenchyma. So it refers to the air tissue. Remember, how many tissue do you have? So do you need to recall your botany lesson back? You have parenchyma, arenchyma, and also the sclerenchyma. Okay, and then the root length of CRDHA and 2O2 significantly increased under aerobic condition, which may attribute it to its aerobic adaptation in terms of water acquisition. So this is the summary of the finding. CR2O2 root length increased. Increase where? Increase in aerobic condition because attributed to aerobic adaptation for water acquisition. Oh, sorry. I need to write this in a much proper manner. Okay. So as you read, take out the keywords, the essence of the sentence. Okay. The photosynthetic rate was significantly higher in CR2O2 compared to BPT5204 under aerobic conditions. So this is very important. Photosynthesis higher to that of BP204. So this guy here has high photosynthesis. Okay, this anatomical and morphophysiological trait associated with the adaptation can be used as screening criteria for phenotyping and selection of genotypes suitable for aerobic system of cultivation. Such study is expected to expedite the development of rice aerobic varieties in aerobic breeding. So this actually the um, discussion slash conclusion for this entire study. So they make comparison between two rice performance, the aerobic and anaerobic rice performance in opposite environment, and then they make a measurements, and then they found out that one variety is more adaptable to one growing condition because of certain adaptation, right? Anatomical adaptation, of course. Okay, so very good that. So abstract is basically just a summary of the entire thing. You can think of it like your novel synopsis. It's done in a very concise manner, right? So you're done with your abstract, good. You got your keywords, and then we move to introduction. Okay, so introduction usually we use present tense. Okay, how I represent a PRS. Okay, I need to keep this. I keep forgetting of my own abbreviations. Rice or Rhiza sativa is grown in non-puddled, non-flooded, fertile soils under aerobic system of cultivation. So that's the statement. Remember, this is your reference. Somebody said this before you, which is in 2001. It's like almost 20 years ago. Since the published time of this paper. Aerobic rice cultivation is gaining popularity in several regions to cope up with water scarcity. A few people say that. These people and these people. So this is how you want to include two different sources of reference. You need to put this semicolon. Okay. The first article that you refer to, you put semicolon and then the second article. And it has to be followed chronologically. This guy here is 2005, and then mention the more latest one, which is in 2009. It is estimated by 2025, 15 out of 75 million hectares of Asia irrigated rice crop will experience water shortage. Okay, and so we just got information from that. Increasing shortage of water is accelerating the adoption of aerobic rice system. Um, wherein up to 45 water system has been reported. So there is a good amount of effort has been done to to use more aerobic rice, okay, because of this water saving, okay, absorption of aerobic rice. So this is something that you need to pick up as you read along. Okay. The response of root growth and development, including efficient uptake of water and nutrient, is an important aspect of understanding aerobic adaptation. So in order to understand aerobic adaptation, the important things are including things like how good the plants in actually do the uptake of water. Because remember, aerobic rice, it's not in flooding condition anymore. At times, maybe the moisture is not the optimum. So the structure of the roots, xylem, and so on must be super optimum. Otherwise, the rice is not going to make it because rice is an annual plant. It doesn't have a very long time to wait for the water to come and then absorb. It has to make do for whatever it's available right at the moment. Okay, the root system architecture, aptly described as the hidden half, critically influences the yield of the crop. The roots are primarily responsible for the adaptation and responses to various stress situations through complex interaction among the genes. A long and thick root system. The ratio of root to shoot weight and root penetration ability of upland rice have been reported to contribute to yield under water deficient condition. So things are important when it comes to how can root adapt to these various ecosystem. Something to do with thick root system, the ratio of root to shoot, and also the root penetration ability. Okay, so these are the things that will determine whether the rice is going to be suitable to be grown in upland or not. Therefore, it's very much essential to study anatomical, morphological, and physiological features of fruits under anaerobic and aerobic condition. So you see that the author here, they make a very general introduction about the rice cultivation system, and then they focus on there is already a movement or tremendous effort to adopt the aerobic rice. So they are giving credits a lot to aerobic rice. Okay, let's do this because this is something that we should be doing. It has so many advantages. Okay, and how can rice be all right in this kind of aerobic condition is because of this root system, thick root, root penetration, and also the ratio of root to shoot weight. Okay, right. And because of this, um, it is very important to understand this discipline first: anatomy, morphology, and also physiology before we decide what variety, or if we want to create new variety, which are more suitable for aerobic conditions. Okay, so creating variety, that's the breeding. Is the breeding being done here? No, but this is the first step of breeding, the platform for breeding. Breeding cannot progress if it doesn't have prior knowledge or um, like you can say fundamental information. So physiology can give you that, anatomy can give you that. So the breeding can be more targeted. There are many different kind of rice variety. Not all of them are going to be suitable for upland or aerobic rice condition, but we can narrow down the selection based on this discipline, right? Okay. So, root anatomy study revealed that traits are responsible for adaptation under different water regime. The number of parenchyma, so that's very important. Number of parenchyma, air cavities, comprised of gas-filled spaces, size of parenchyma. Is it parenchyma or parenchyma? Doesn't matter. You one. I think one is British, one is American. Um, uh, people know what you're talking about. Okay. And the roots are influenced by the diverse water regime, viz flooding, um, anaerobic and hypoxia condition. Okay, sometimes in science paper, they like to use this kind of word, viz. What is that? Certainly this is Latin. Um, this is Latin. This actually means namely, or that, oops, sorry, or that is, viz. Um, the full word for this is, what is it? I think it's called vidalicet. Okay, it means namely, or that is. Flooding, anaerobic or hypoxic condition, parenchyma contributes to the transport of essential gases from the above ground parts to the roots. So yeah, parenchyma is very important. So from here, you know that rice are able to live in submerged water condition because of this parenchyma tissue. So parenchyma tissue, you do not have any um, content in it. It's just gases. Okay, so even though the roots is completely live in a flooding condition, but as long as the roots got parenchyma, the gas can come from the top part. Okay, the shoot part, and then the gas can come down and then nourish the underground rooting network. The supply of oxygen from roots to shoot is facilitated by the formation of parenchyma, which is essential of plant underwater logging condition. See, what the thing I said is just been said here as well. Okay, parenchyma quantification is essential to know the presence of parenchyma formation in the rest genotype under different water regime condition, the number and size and so on of the parenchyma to be associated with drought tolerance. So it's very important to have this parenchyma for rice. Certain anatomical changes in rice root, the cell wall thickening, lignification, the axonomes, endodermis, and sclerenchyma has also been observed under aerobic condition. So beside all the parenchyma thing, under aerobic condition, there are other things that are also changing for the rice to be able to grow aerobically. These are not very prominent, but they still happen. So it's worth mentioning here. Okay. The morphophysiological trait is important in this relation to aerobic adaptation. Okay. There are already several reports on yield, morphology adaptation, different anaerobic and aerobic system cultivation, but sparse information is available on the physiological trait conferring aerobic adaptation. Yeah. So the author is actually highlighting. Oh my God, sorry about that. Is highlighting in the science world now, what is lacking? People do not talk about so much about what. Yeah, so these are the things that people don't talk about. So when you do your experiment, you can think of your experiment as to be a confirmative experiment. You want to confirm the finding of previous study, because remember, any study should be reproducible. If you can confirm the study, then that means the scientist that the study originally came from has done a very good job. But if the study is somewhat not reproducible, you got completely opposite results, then the study can be questionable. There are many factors, but things are not yet written in stone. Okay, Arabic rice cultivation largely depend on the initial plant establishment. So plant establishment is very important. Okay, and so they also tell you, the author here, they tell you what they did, okay, to come up with this. So can you yes, you can mention this in the introduction. This like I like to call it like a monologue monologue sentence. Okay, they kind of talking to themselves, and then they want people to know, okay, actually this is what we did, right? Okay, so after you're done with the introduction, then you go to the materials and methods. Okay, let's see. Material methods, usually we use the past tense. Okay, we use the past tense. Unless if you are referring to something like a regular statement, then use the present tense, okay? So this experiment used rice, two genotypes, which is the BPT 5204. Remember, this is anaerobic, and also CR done to 202, which is an aerobic adapted. Okay, and they also described how the condition it was done. Um, there's a base of irrigation applied when required to provide to maintain aerobic conditions. You see, even though it's aerobic condition, they still water the plants. Okay, just to maintain enough. It's not flooding. Okay. However, for the anaerobic, they maintain the 2 to 5 cm of water for a period of 100 and 220 days in the poly house. Poly. What do they mean by poly house? Poly house. This usually referring to different kinds of polymer or substance. It can be polyethylene, polypropylene. Yeah, it's just a general term for that. Okay, and they also said that the procedure followed were essentially as described earlier. So whatever they did here, they didn't go into detail, because if you want to read detail, you can refer to these people, this author, previous author. This has been done before. Okay, which is a confirmative as well. They repeated what these previous authors did, and apparently, yeah, your method is good in growing rice, so we can confirm that. See, you just did a confirmative study, even though you didn't have intention to do so. The genotype were grown in the polythene bags containing 15 kilograms soil under aerobic and anaerobic condition in the greenhouse. So figure 1. So how it's done, they also put it in the figure. Yep. So this is how they conducted the experiment in the poly house, and then they have the rice grown in these poly bags. Okay. Yeah. So obviously this rice, I think this should be, yeah, A and B, which is here, A, B here. So A is the aerobic and B is the anaerobic. You see, it's not flooding, no flood. Okay, and then this is a flood, right? Okay, so let's see what else did they do. Okay, they also say that the root tissue samples harvested at panicle initiation stage, about 60 to 70 days after germination. So this actually tell you this experiment is somewhat destructive, because when the plants are about 70 days old, they removed from the soil so that they can get the parameters for the roots. Okay, and in 3 independent biological replicates from each genotype per treatment. Say they also mentioned the replicates. They mentioned how many replicates, which is 3 for each genotype per treatment. The morphology physiology parameter from root and shoot were recorded at panicle insertion in 3 diff replication for 3 seasons. Okay, so so for the morphology and physiology, and this above here, this is for the agronomy, which is the root agronomy. And for the morphology and physiology, also they did in 3 replications. So this is how you report your own finding. Okay, for 3 seasons. Okay, and I think this is the person responsible for this, how it's done. Okay, right. What else? Okay, they have covered the plant material and treatments, and then comparative root anatomy. Okay, so what did they do with the roots? So the root tissue of the varieties at panicle initiation they cut into thin sections in water using short surgical blades. So this is where I said before, you need to be very good with your imagination. You need to picture as you read what the scientist did. Okay, this is what we call as active reading. Active reading simply means you picture as you read and then you make connection. Turn the still images in your head into a movie. Okay, and then make you are the director of this movie. So create a story visually in your head. Okay, how it's done? Well, I can't really teach you that. That depends on how crazy your brain is. The crazier, the better. The section was stained using acridine orange. So they say that after they have made the section, they stain using acridine orange. This is a histological stain. Why they stain? To So that parts of the structure, microstructure of the roots can be distinguished later. Observe at 10 and 20, 10 and 40 times magnification under a simple light microscope. So this is the name of microscope that they use. Okay. Okay. Detailed comparative anatomy study was carried out using scanning electron microscope. So they did use the regular light microscope, and also they use this SEM, scanning electron microscope, as well. So they use two kinds of microscope. Very useful. And then the root tips from crown roots were cut using ultramicrotome, and 10 micrometer root section was studied in 3 replicates. The root section were visualized using the scanning electron microscope at 120 times magnification, and the vascular bundle, the stellar region, were observed at 3300 magnification. Root section images were captured using a digital camera connected to a microscope. Okay, here or where you can improve. Digital camera to a microscope. So it's good to tell what camera, okay? Because camera has different ability. Some camera can see better, some camera can see less than optimum. So this is something can be improved, okay? All right, and then the root anatomical parameters like parenchyma, cell number, cross section. See, not only that they mention the parameters, they actually also state what is the unit. So they are very particular about this. Vascular bundle, okay, and phloem number, xylem area. Area, maybe they miss something. This area should be in micro square micrometer, okay, and root thickness, and recorded for comparative analysis. Okay, so more or less we can have a short movie of how they did this. Okay, morphological observation. The entire plants were carefully removed by cutting the polythene bag without damaging the roots. Okay, the roots were washed by dislodging all aggregate soil using a high pressure water pump. Shoot roots were cut apart at the node interface. Root and shoot morphological observations like root length, shoot length, total length, height, and so on were recorded. 3 plants per replication. So these are all the parameters that they recorded and measured. And how many times they did? They did for 3 plants per replication. Okay, what about the physiological observations? So they use 50% of flowering stage. So this is very important, meaning that is there any rice? No rice yet, just the flower, which is only 50%. This is important because this is the indication that they did physiological measurements at early reproductive phase of rice. Okay, that's why it's also said in addition to picture and create a movie as you read, you also need prerequisite knowledge and information about rice. Okay, if you have taken the rice class before, or maybe you have grown rice before, then you will know this. Okay, what equipment they use? So they use this portable photosynthesis system from Lincoln, Nebraska, USA. So this is a very common photosynthesis machine. So what did they measure? They measured net photosynthesis rate. So this is the unit, transpiration rate, and also the stomatal conductance. Okay, stomatal conductance. Okay, intercellular CO2 concentration, and water use efficiency and intrinsic water use efficiency. Okay, this thing, however, I'll tell you ahead of time, water use efficiency and intrinsic water use efficiency, this is not measured, okay? This is the thing that they should mention here is this is calculated, because these parameter, water use efficiency and intrinsic water use efficiency, this is a ratio. After that, leaf chlorophyll content was estimated using fresh flag leaf sample. So flag leaf. Flag leaf means that, so this is very important, they use last leaf before panicle. Okay, using SPAD, this is the equipment. So it's described here. What is it? A portable convenient meter used for estimation of leaf chlorophyll content. And then secondly, for the pigment quantification as well, they took 100 milligram of the flag leaf, they cut into small pieces and incubated in the solvent mixture of 80% acetate for 48 hours. Yeah, so this, I think, you might want to pay attention, because we can do this tomorrow. Yay! Yeah, so just a sneak peek for your lab. So we are going to do the pigments quantification. Yeah, so we're going to do this. We'll talk more about this later. And then they told how they came about with the calculation of the chlorophyll. So this is the equation that they use from this author. Okay, Uh, so the SPAD chlorophyll meter reading were measured for selected flat leaf. So they also mentioned the position of the leaf, where they use the top, middle, and lower. This SPAD machine is like a clip. Okay, you clip this equipment on the leaf, and then the equipment will tell you the relative amount of chlorophyll. Okay, however, this equipment doesn't tell you the specific amount of each chlorophyll. It's just a general amount of total chlorophyll. If you use, that's why they did other method. They do the acetone method here. You see, they use the acetone method here because they want to quantify for specific chlorophyll, namely chlorophyll A, and also chlorophyll B, and also total chlorophyll in milligram per gram. Okay, so this method, the acetone method here, not only can quantify specific amount of chlorophyll, but also with the unit. So this is very quantitative. This chlorophyll spectrometer, it's a relative value. So this is like partial quantitative. Okay, so in a form, in a way, if you have a SPECT reading, it can validate your biochemical extraction method as well. If they look the same, then you are doing the right thing. Okay, and finally, after they are done with this, they did the statistical analysis. They say here what software they use, version what, okay, and then what kind of statistical test that they use. They use the two-way ANOVA and also the LSD analysis, okay, at 5% of alpha level, okay, or the level of p-value, as you remember from your statistic class. Okay, you're done with the um, materials and method. Now, here come the result and discussion. Okay, result and discussion, some journal, they combine, like this one. Many other journal, they separate. They have a separate section for result, separate section for discussion. Okay, so result is usually in past tense. Past tense. Discussion is a combination in general. Okay, because this paper combine result and discussion all in one go, you're going to see past, present, and future tense being used interchange from time to time. So there's no specific arrangement to that. Okay, let's see. How do you detect whether it's a result or discussion? So they divide their section into subsection. So this is a subsection. So the first subsection is talking about the comparative root anatomical study. The comparative root anatomical studies on the high aerobic and anaerobic condition aids in associating the adaptation to root morphological traits. Is this a result or discussion? This is actually a general statement. You can call it, this is a pre-discussion. Xylem area and root thickness were significantly p-value less than 0.5 higher under aerobic condition as compared to anaerobic condition. Okay, so they have started the result. How do you know? Result past 10, right? This is a past 10. Higher under aerobic condition. So it's comparing. Significant differences in parenchyma cell size and number were observed under simple microscope. So this is a result. Parenchyma cell number, vascular bundle, diameter, area, root thickness were significantly higher in CR202 than BPT504 under aerobic condition. Okay. All right. So this is the information that you get. This variety here have all of these parameter higher than the other variety. In BPT5204, parenchyma cell number, radius and cross-section, vascular bundle, diameter, xylem area were significantly higher. So to look at this, you need to refer to table 1 and figure 2. Sorry, let's move a bit down here. Okay, so this is a table 1. All right, so look at this. Pretty much this is how you do as well for your own study. So you have your genotype, the species that you have, BPT5204 and CRTHN. So they did the study for this thing too. Yeah, let's divide that. Okay, so you have these two grown in one environment, aerobic condition, and these two you grown in anaerobic condition. So the parameters are how many here? 1, 2, 3, 4, 5, 6. There are 6 parameters here. Okay, they also said that they did the ANOVA. Okay, not only that they did ANOVA, they also did the LSD, the least significant difference. So this is the post hoc for ANOVA. Post hoc analysis. ANOVA do not tell you which treatment is different. ANOVA only tell you among the things that you tested, somebody is different from other people. Just that. That's why we need to do post hoc analysis, like LSD. LSD, there are many type of post hoc analysis, not postdoc, post hoc analysis. One of them is LSD. Post hoc analysis will tell you specifically which one is different using this thing. You see the letters here, small letter here. So this is what we call as letter grouping. We read it according to the column, okay, downward here. and it's true for other parameters as well. So we have the letter grouping here as DCAB. Okay, so what's the difference from A to D? So you can quickly tell your head that A belongs to the highest value, the grouping for the highest value. D is the lowest. And this is not a general statement, okay? It depends on your software. It depends on how you arrange your observation. Sometimes this can be the other way around. So that's why I like to do it this way, so that I can know the moment I see here, oh, BPT anaerobic is actually different, significantly different, because they have a different letter. When you have different letter, they are significantly different. But to what degree? Oh, this guy got A. We know that A belongs to the high group, and D belongs to the low group. So you can quickly create that mental visualization again in your head. Who has more, who has less? Okay, and the thing also applicable to other parameters. Okay, so the radius, you can see that so remember, A is high, whatever after that is lower. So we can see that, okay, apparently this CRDHN, when you grow in aerobic condition, is it going to be high or low? So it's consistently higher. See, this is on 26. It's consistently higher, consistently higher, consistently higher, consistently higher when compared to the BPT variety when it's grown in under aerobic. Clearly, this CRDHAN is, it's true, it has been adapted to aerobic condition. What about the BPT? BPT has been adapted to anaerobic condition. You can see that for BPT, it is the one which is consistently higher for each of the parameter. Okay, but not this one. This one is higher and not this one. So there are two things which are different here. BPT under anaerobic condition, it is still lowest when it comes to phloem number and also the root thickness. So this is your observation. Not only that you are making observation and interpreting this data, but you are trying to make sense. Oh, this variety, even though it is adapted for so long, it's still becoming this way when you grow it in opposite environment, things like that. Okay, so science it takes time. So however, you can be assured that the more you do this, the more skillful you are going to get in terms of interpreting and connecting the dots. Okay. All right. Aeroncama in roads provide transport of essential gases from the above ground part to the road. So this is discussion. So this is discussion. Our result for Aeroncama cell number were consistent with Suralta, Henry, and so on. And also this is, they are comparing. So this is actually discussion. They are comparing with the previous authors, whether they agreed or not. Okay. Aeroncama formation was up to a lesser extent on the drought condition than under the flood condition in both aerobic adapted and lowland genotypes. All right. Okay. So this author, Kundur et al. In 2015 revealed highly significant variance among diverse genotype of 20 days old seedling for Aeroncama cell number and xylem cross section. So this is something that you need to take time and also to make some sense about it. You see, results, pure results, it will not mention other people. pure result is like this. Like this sentence here. Do you see it's comparing to anybody? It's not quoting or referencing to anybody. It's just simply stating the obvious. When it's clearly stating the obvious, this is a pure result. When it's trying to make a sense or comparing, agreeing or disagreeing, like what we have in this paragraph here, this is discussion. Okay. So, and then what it says, our results suggest that the formation of parenchyma being a constitutive process, fewer parenchyma must have formed under aerobic condition as compared to the anaerobic condition as essential anatomical adaptive trait for survival under aerobic condition in rice. So this is their finding. The rice need to have a good parenchyma, but it has to be fewer under aerobic condition. Okay, which is true. You do not need lots of gases in your body if you are already not submerging in water. Why waste the time? Why waste the structure, right? Make it less so that you can reinvesting the resources into making something else. Okay, right. Okay, they also include a image here. So this is the distribution of an image. Rice root anatomy at panicle initiation, stage under aerobic and anaerobic condition. Okay, so this is the genotype. This is what we call as micrograph. This thing here. This kind of image, we call it micrograph of scanning electron microscopy. All right. So this is what it looks like. You're going to see this is BPT 5204, what it looks like in anaerobic condition, what it looks like in aerobic condition, and then genotype. Look at here. I think they made a mistake here. It shouldn't be 2 times. So I think this should be the CRDHAN 202, right? It should be the other variety. So it's a typo there. It's all right. We if you are presenting things like this in a journal club, this is things that you highlight, okay, to make it more refined over the time. Okay. So in this electron microscopy, no color, okay. So you get your you can have put a label. So the label here. So this is the root hair. This is the parenchyma cell. You see, this is empty. Empty, nothing much. So they got the pericycle, got the xylem here, and you got the phloem tissue here. Okay, right. And you're going to see this. You see this? The bar here. So the bar here, actually what we call as the scale bar. For the length of this bar on this image now, it's going to be equivalent to 100 micrometers. So if you move from here to here, the same as the length of this bar here, this is 100 micrometer. So this is very small. This is not even 1 millimeter. Okay, 100 micrometer is 1/10 of millimeter. 1 millimeter is super small already. This is even smaller than that. Okay. Okay. What else they do? Okay. So this is the cortical parenchyma. So it's good. You see, the parenchyma is actually the ground tissue. Okay. Ground tissue. And then you got the cortical fib, which is the sclerenchyma. Sclerenchyma. So this is the usually the more fibrous one. Fibrous, hard. Okay, right. And they did some kind of measurement as well. See, this is the measurement here. Although I'm not sure what kind of measurement, so it will be useful to describe. Okay. Yeah. Just from the perspective of a microscopist, okay. Um, I was a microscopist before. If I were to present this in a journal club, this paper, maybe I would have more comment on how this is done. Okay, I would um, scrutinize the way they did this electron microscopy. Okay, because let's say what they did with the electron microscopy method. Okay, so there is a lacking information here actually. They didn't tell no method to prepare sample for electron microscopy. There is no, yeah, they didn't tell what is done. They described in elaborate manner what they did with the light microscopy, which I kind of feel weird why they don't include the image from the light microscopy. They did the electron microscope, but they didn't tell what it's done, because light microscopy, the method is much more laborious. Okay, more things to be done. So this is something that actually it's not optimum. If I were them, there are things that I would do differently to make this look more how do you say, fresher looking. You can see, can you see all this crumpling looking, like a peppery, like a crumpled paper of the tissue here? Actually, this can be avoided. Okay, this is the sign of tissue collapse. Tissue collapse. Very often when this is done, it's not properly dehydrated. Okay, yeah, yeah. But that's beside the point. That's just to tell you, when we read journal, not only that we scrutinize the text and the table, but we also look at the figure, and then we highlight, as someone who has done it before, it shouldn't look like this. It can be better, yeah, so that you can have a good visualization of your specimen. Okay, all right. Okay, and this is another subsection. Okay, right. Okay, what's the time now? I don't want to go too overboard. Okay, so pretty much they just did what they did before. They just they combined the discussion and also the result. And the take-home message here is here. Oh, they put it in this side way. Let's have a look. What exactly happened here? Um, yeah, I have actually turned it the other way around. Why it's not opening? Mm, let's see. Why is not opening? Okay, so this is the table just now. Okay, it's. I feel weird as well. Why they put it this way? But anyway, let's turn it. Yeah. So this is the results for the second subsection. Okay, so you have the root length, shoot length, total plant height, and so on. There's so many things. So what's important here is we can see that for the anaerobic, oh sorry, for the aerobic, between the two variety, who is higher? Let's see who is higher. This guy here, 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 this guy, this guy, this guy, this guy, this guy, this guy, this guy. So it's not as consistent as the the previous observation for the anatomical traits. Okay, for the parenchyma, the vascular bundle properties, pretty much the CR202 is always the higher number compared to the BPT 5204 when it's grown in aerobic condition. But for the root morphology, the detailed part of the root morphology as well as the root shoot properties, you can see that it's not a similar pattern. Some parameters, BPT variety, it's has higher reading when it's grown in aerobic condition. Okay, but is it significant? It's higher is one thing. Is it significant? No, not very significant because the letters are still similar. What about here? Is it significant? Not very significant because the letter is still similar. Yes, this is significant and not very significant. Okay, so we can tell that for aerobic adapted variety, they have longer root. Well, why? Because it needs to penetrate deeper to the soil. It's not in flooding condition. You need to the plant needs to look for more water. Shoot length, total length, and for the root fresh weight, they're similar. When it's okay, when it's not significantly different, we call this as they are similar. Okay, they are similar. Similar, statistically similar. Statistically similar. Okay, and you can call this as significantly different. Okay, yeah. What about the total dry weight? Total dry weight of everything. Yeah, the CR202 variety is heavier. Yeah, but if you want to compare with the anaerobic situation, um, they are not that different when it comes to total weight. Because in anaerobic condition, the plants have to worry less about because water is always there, so they can just do the photosynthesis, do the transpiration for as much as they want. Okay, right. So is it true? These things, CR variety is actually have longer roots. So this is what the finding here. Okay, so CR variety has a longer root in aerobic condition, longer root, but um, for the root shoot ratio, this ratio here, it seems like the BPT variety has a bit more. Can this be proven? So let's see what they give us. They also give us the figure. Yep, the authors give us the figure as well. So you can see that under aerobic condition, the BPT variety and aerobic condition, yeah, the aerobic condition for the CR DHAN, can you see the root here? It's all the way here. So it's a bit longer than this aerobic condition. Yeah, but when both varieties grown in anaerobic condition, you can see that not only that the roots, all roots kind of the similar length, but the BPT, which is anaerobic type, has more root mass. You see, the root looks massive compared to this root here, which looks pretty much less. Okay, so meaning that um, for aerobic condition, actually, you need to have a long root. Okay, the long root is more is more important when you want to grow the rice in aerobic condition. Why do you think the rice has massive roots in anaerobic condition? Remember, okay, this actually water. Why? Because you can relate back with this root um, electron microscope image that we saw earlier. You see, this is parenchyma. Parenchyma means there's lots of oxygen. There's a lot of oxygen. There's a air there, oxygen. Since this is underwater, more roots, not longer roots, okay? More roots equals more parenchyma equals to more oxygen. So that's why. That's why anaerobic rice, they have massive rooting system. Is it to absorb more water? No, it's not to absorb more water. They want more of this parenchyma so that they have more oxygen. Okay, that can come from the top part. What about the anaerobic uh, CRDHN? They too increase the root mass. You see, this root mass, even though this is smaller than this BPT variety, it is still bigger. Okay, you see, very little here compared to here. So we can know that um, for aerobic condition, root no need to be massive. Why? No need for parenchyma. Because it's not in flooded in a submerged condition. The root can breathe on its own. Can breathe easily. Okay, so that's how you make the connection and make the discussion. Okay, and whatever I said, I'm sure they have mentioned somewhere in the text. Okay, we don't have time to go to everything, but pretty much what I said here has been mentioned in the text. Okay, all right. So what about the photosynthetic response? Okay, and for this, okay, this is a table, the result here. You can see that the Cr2O2 has the higher photosynthesis. Okay, however, but when both is grown using the anaerobic condition, they are similar. Okay, what about the stomatal conductance? Okay, stomatal conductance is just the passage of substance in, out. So the machine measures how much water is able to go in and out of the stomata, and that's why the unit like this: mole of um, water per square meter per second. Okay, we under aerobic condition, they are not that different, and as well as anaerobic condition, they are not that different. Okay, so meaning that stomata is not something that can be greatly affected by growing in either an aerobic or anaerobic condition. What about the intercellular CO2? Intercellular CO2 simply means the content of CO2 in between cells, not inside the cells, in between cells. Okay, and yes, you for the CR, you have higher intercellular CO2. When you have high intercellular CO2, meaning that your plants have more CO2 to do photosynthesis. And this is actually translated here. not here, here. This is higher, right? CR variety is higher. Why is it higher? This is how you explain. This is your finding. You find out that photosynthesis is higher. Why? Apparently, the intercellular CO2 for the CR variety is higher, significantly higher than the BPT variety. And why? Why this is higher? Potentially because stomata can let higher passage of gas. Okay, this is not significant, but it is still higher, 0.16 compared to 0.1. Okay, transpiration rate. Transpiration rate actually is quite similar to stomatal conductance, but it doesn't tell you about the stomatal properties in the equation. No stomata. It just tell you about the general amount of water leaving the plant without having any respect on the stomata properties. You know, sometimes stomata have their impart some kind of restriction to the gas exchange. Okay, but transpiration do not include that restriction in the equation. Okay, and then we got a water efficiency, and this is just a ratio. That's why you see it's written this way, the unit here, and it's photosynthesis divide by respiration, transpiration. Sorry, photosynthesis divide by transpiration, and intrinsic water use efficiency. Okay, this is incorrect for this. That's the way they report here. This is not the unit here. No, intrinsic water use efficiency also ratio, but photosynthesis over stomatal conductance here. Okay, this guy here divide by this guy, you got water use efficiency. So what's the difference between the two? This thing, no stomata. This thing take into account stomata. Okay, right. And finally, the leaf chlorophyll content. You can see that they're pretty much similar actually. That looks similar. That looks similar. What about the chlorophyll content? It also looks similar. Total chlorophyll, yeah, also look kind of similar. So nothing much going on. So you can see that water availability don't really have impact on the chlorophyll A or chlorophyll B. So they kind of similar. And this is the SPECT reading as well. So this is a to double confirm. So this is the acetone method, biochemical procedure to extract each chlorophyll type. And then this is just a this is just the reading from a machine. And also for the machine, they are not different. So aerobic, anaerobic condition do not have any impact on the um, nitrogen content and also the chlorophyll content in the plant. Okay. All right. And finally, you have your conclusion here. So it says here, the present investigation demonstrated that root anatomical traits, that is fewer parenchyma formation, thickened roots, larger xylem area, and the morphophysiological parameters such as root length, total dry weight, and photosynthesis rate are critically associated with aerobic adaptation. So all these parameters here associated with aerobic adaptation. It was observed that the adaptation in terms of deeper root length in order to acquire and take up the water from the soil operates under aerobic condition. This anatomical morphophysiological parameter can form the preliminary basis of root phenotyping for the development of suitable rice genotype for aerobic adaptation. So they are giving potential, and also they are giving the suggestion. So this is what it should. So they're doing it the right way here. Conclusion here. Their finding have this potential as a initial knowledge for breeding program, and also a suggestion how it's to be done based on the parameters that they have taken. Okay. However, they also put a disclaimer. These observations need to be further tested in more number of genotype adapted for aerobic and anaerobic cultivation. So when they say this, maybe you can test this on your Malaysian variety because these are Indian variety, you see, from India. All right. And finally, they put all the references that they have here. Okay. And sometimes people put here. So this part, we call it acknowledgement. Okay. Acknowledgement. Just a courtesy and so on. And then finally, the references and all the references. Okay. That's all for this paper. Yeah. I will give you the activity for you later. It's wait, that's fine. Maybe I can give it to you today. So you need to do presentation on the final week, week 14. And for your presentation, we are going to have like a journal club kind of presentation. So I have introduced you to this kind of scientific paper. Now, what I want you to do is, why don't you go to, you don't have to anything to just now, for now. Go to Google, and not Google, go to, what do you call that? YouTube. Have a look at how people do the journal club presentation. I can give you the link here. See how it's done. And then I want each group, for example, I got two papers here. Yeah, that looks good. That looks good. Okay. Group, this is what? This is about, this is a very short paper. So it's good. Yeah. So group top, why don't you take this? Can I save this? No, I don't want to save this. Let me try to save it as PDF. Yay. So group top, you take this paper about corn, how it's grown in different ways. So I'll just put your group. And then group bottom, why don't you take this paper? I'll save this as well. Oh, don't tell me it asked me to. Okay. Okay, so that's fine. It asked me to the PDF. I want, let me see, can I get into it without signing in? Okay, I'll just put print it as a PDF. That's right, that's fine. So group bottom, why don't you take this paper? Okay, what do you need to with this paper? So each of you work in a group, in your group, read everything about this paper. It's pretty much like the paper that I just shared with you in terms of the component and the way it's written. And then go to YouTube, see how people do the journal club presentation. Do it in such a way that the lay people can understand it. Okay, but here's the trick. I want you to make the presentation. It doesn't have to be long. Maybe like about 15 minutes only. I want you to make it. This is very scientific, right? This is very scientific. I want you to make it in a very way even people in the local grocery store can understand what this is all about. So turn this into presentation. You can do rewording, you can do rephrasing, you can add additional images. You see this picture, this paper here, it talks about a corn, right? There's not a picture about the corn plant or corn cobs. So you can be more creative about that. I want the presentation to be delivered. Well, we have been doing English all the time, right? Let's do this one in Malay. You present to me in such a way. Uh, I think we can do live. I'll think about it. I'll think about it. Um, we'll do it in Malay so that the rest of Rakyat Malaysia can understand what this paper is talking about. Because this is a very interesting skill to have. You read scientific paper, you understand scientific paper, and then you are able to tell the finding in the scientific paper so that the poor farmers can get benefit from the finding. How do you do it? All right. So I'll give you your paper here. Group to group bottom. Let's see. You can get from the chat the paper. All right. So and then find it into and look how people present the journal club. It doesn't have to be exactly the same. Okay, again, it's pretty much like your ebook. Oh, your ebook. I will return to you after. I need to read a bit more. Your ebook. Be creative about it. Try to present. You want to use a PowerPoint. You want to use Canva. Whatever. You can use whatever you want. Present it in such a way that Rakyat Malaysia in general can understand. Remember, this is in English. You need to present in Bahasa Melayu. Okay, so that's a skill. You in Malaysia, you cannot be a good scientist if you are very good in reading scientific papers and so on, but you fail to explain to a poor pakcik at the field how to apply nitrogen fertilizer. Okay, so let's develop your skill. All right. Okay. Uh, yeah. Okay. This is for the group bottom. Right. It's the same story. You can. There's no picture. This is about what? I think it's about chili. Yeah, this is about chili. No picture, nothing. You can include on your own. It doesn't matter. Yeah, as long as the message arrive to the audience. All right. Okay. I think that's all for today. Um. Yeah. Uh. I'll be around until 9:37 for your questions. So yeah. Any question you can ask me. Okay. Okay. I think if you do not have any question, we can dismiss the class. So I'll see you tomorrow um, um, at the lab. We're going to do some biochemical work. So please bring your lab coat, and I'll see you then. Please don't be late. Okay. So thank you for coming. Bye.
Thank you, Ratan.
Welcome.